Hey there, science fans! Today we're diving into something super cool. It's called UV Visible Spectroscopy. That's a big name, I know. But trust me, it's all about light and colors, and how we can use them to understand stuff. It's like seeing the world in a whole new way. Science is all about looking closer, isn't it? Let's get started on this amazing journey. So what does UV visible even mean? Well, UV stands for ultraviolet light. That's the kind of light that can give you a sunburn. We can't see it with our eyes, but it's there. Visible light is the light we see. Think of all the colors in a rainbow: red, orange, yellow, green, blue, indigo, violet. This science uses both kinds of light. It's a full spectrum of fun. Now, what about spectroscopy? That sounds like something out of a spy movie. But it's just a science word. It means studying how light and matter interact. Matter is just stuff, anything and everything, like the juice you drink or the water in a lake. Spectroscopy helps us see what happens when light hits that stuff. Does it bounce off? Does it go through? Does some of it get soaked up? That's the key. Why is this whole thing so neat? Because UV visible spectroscopy lets us identify things, and it can tell us how much of something is there. Imagine you have a mystery liquid. This science can help figure out what's in it. It's like having superpowered vision for chemicals. We can learn so much just by shining a light. Isn't that incredible? Science rules! Okay, let's get down to the nitty-gritty. How does this UV visible spectroscopy actually work? Imagine you have a glass of cherry juice. It looks red, right? If you shine a white light through it, what happens? Some light passes through, but the juice soaks up or absorbs some of the light. Especially the colors that aren't red. That's why it looks red to us. Now, different things, different molecules absorb different light. Think of it like this. Every molecule has its favorite colors. Some molecules love to absorb UV light. Others prefer to absorb green light or blue light. It's like they have a unique appetite for light. This is super important for scientists because it means different substances will affect light differently. This difference is what a UV vis machine measures. It's like a unique light signature. The UV vis machine is very clever. It shines light, one specific color or wavelength at a time, through a sample. The sample is usually a liquid in a small, clear container. This container is often called a cuvette. The machine then measures how much of that light makes it through, and how much of it got absorbed by the sample. It does this for many different colors across the UV and visible range. This gives us a lot of data points to look at. From this data, we get something called an absorption spectrum. It's like a graph, or a special kind of fingerprint. This fingerprint shows which colors of light the sample absorbed most, and which ones it absorbed least. Since different substances have unique absorption fingerprints, scientists can look at the spectrum and say, aha, this sample contains molecule X, or there's this much of substance Y in here. Pretty amazing, right? Pure science magic. So what's this amazing machine called? It's a UV visible spectrophotometer. That's a mouthful, but it's a powerful tool. Think of it as a high-tech detective for light. It has several key parts working together. Each part has a very important job to do. Let's peek inside this science gadget. It's less complicated than it sounds, I promise. There are usually 4 main heroes in this machine. First, you need a light source. This isn't just any old light bulb. It needs to produce both ultraviolet and visible light. So often there are 2 lamps, one for UV light, like a deuterium lamp, and one for visible light, like a tungsten lamp. This ensures we can test the sample with a whole range of light, from the invisible UV to all the colors we see. More light means more information. Next up is a very cool part called a monochromator. Mono means one and chroma means color. So it picks out one color or wavelength of light at a time. It takes the white light from the source and separates it into all its different colors, like a prism making a rainbow. Groovy! Then it lets only a tiny sliver of one specific color pass through. This way, we can test the sample with pure single colors. Then, there's the sample holder. This is where our mystery liquid goes. Usually it's in that special clear container, the cuvette. The beam of single-colored light from the monochromator shines right through it. The cuvette has to be made of special material that doesn't absorb the light itself, like quartz for UV light or special glass for visible light. So we only measure what the sample does to the light. Accuracy is key in science. And finally, the detector. This is the part that sees the light. It measures how much light made it through the sample. It's super sensitive to light intensity. It compares the light that went the sample to the light that came of the sample. The difference tells us how much light was absorbed. This information is then sent to a computer, which shows us that awesome absorption spectrum graph. Science. So, why is this UV visible spectroscopy so important? Well, it's used in tons of different science fields. Chemists use it all the time in their labs. They can use it to check the purity of chemicals, or to find out the concentration of a solution. Imagine making sure your drinking water is safe. UV-Vis can help detect certain pollutants in water. That's science helping keep us healthy. It's a reliable and well-understood technique. It's also a big deal in medicine and biology. Think about analyzing samples from patients, like blood or urine. UV-Vis can help. It can measure things like proteins or DNA, or check for certain substances that might indicate illness. Doctors and medical researchers rely on these results. It helps them understand what's happening inside our bodies. Making new medicines? UV-Vis plays a role there too. It helps ensure the medicine has the right active ingredients. Food science uses it too. Want to know the quality of olive oil? Or the amount of certain nutrients in food? UV-Vis can help analyze food products. It ensures quality and can even detect spoilage, even in manufacturing, making dyes or inks. UV-Vis helps make sure the colors are just right. It's a versatile tool for many industries, from the lab bench to the factory floor. So, to sum it all up, UV visible spectroscopy is awesome. It uses the power of light, both seen and unseen, to tell us what's in a substance and how much. It has a cool machine with a light, a color picker, and a detector. And it's super important in chemistry, medicine, and so much more. Knowing about UV-Vis helps us see how scientists explore our world. It's a fantastic example of science in full color. Keep asking questions. Keep exploring. Science is everywhere.